This is Diploma 1 from the Institute of Brewing, and we're covering the first section on barley. There are two varieties of barley commonly grown for brewing: two-row barley, where the ears are counteropposed, and six-row barley, where there is a swirl of barley grain around the one site. If we look at barley in general, all barley is six-row, but in the two-row barley, four of the ears here don't develop, and they're called vestigial. So we have small ears here that don't develop, and the main grain here develops. In six-row barley, all six ears develop, and we get a swirl around the centre of the barley corn. Six-row barley are commonly used in North America for brewing, whereas two-row barley is most usually used in Australia and Europe as the main source of malting barley. If we look at the differences between them, six-row barley give a better yield per acre. Two-row barley, as we said before, have only two grains that develop, and four are sterile lateral florets. In six-row barley, because there's so much pressure around the grain, we see that some of the grains are slightly asymmetrical, where they abut up to the other grain in the cereal. Six-row barley tend to generally have higher nitrogen, lower extract, but better enzyme potential. This makes them particularly suitable for brewing with high adjunct ratios, because they have plenty of enzymic power to help convert the additional extract. In the UK and most of Europe, all the malt is made from two-row barley, but as I said before, in North America it's common to use some six-row barley, particularly for the larger scale brewers who use higher levels of adjunct. As well as the difference between six-row and two-row, varieties can also be planted at different seasons, particularly in Central Europe. In certain areas, winter barley is planted in September, October. It grows up through the soil and sort of overwinters, growing very, very slowly, and then is ready for harvesting in mid-July, whereas we can late plant barley in March and April, and it is a faster growing variety, and then it is available for harvesting in August. If we look at the differences, winter barley is sown late in autumn. It grows very slowly over the winter. It has to be frost resistant, because obviously it will be killed. We may need more disease control on it because it's in the soil for longer. And in the UK, it's harvested in July in England, and because it's slightly cooler, later in August in Scotland. And it tends to have a good yield of about 8 tonnes per hectare, provided we have some form of controlling fungi growth. The spring varieties are sown in March and April. They have a shorter growing period. Early ripening is important, i.e. they have got to grow faster in a shorter period of time. They tend to be harvested in early August, and the yield is slightly lower at 6.8 tonnes. Another feature of barley is its endosperm. This is the starchy store in the centre of the grain, and the endosperm is surrounded by beta-glucan cell walls and has protein in there. So the protein matrix varies according to growth conditions, lots of fertilizer and variety, and we particularly focus on two types of grain: mealy grain, which is open and easy for water penetration and enzyme transfer, and steely, which tends to be much harder and more dense, and because water can't flow through it so easily, it makes it a lot harder for modification. If we look at the electron micrographs, here's a mealy grain with plenty of open spaces. You can see how water and product could flow through it and break down these beta-glucan cell walls, which is one of the principal functions during malting. And typically these barleys are low in nitrogen. In contrast, this is a steely grain which would tend not to be suitable for malting at all, but used as a feed barley. And here it's very densely packed. You could imagine that water flow through this grain would be very difficult, and there are low air spaces, and so the passage of enzymes and water through the grain is much slower, and hence it doesn't modify so well, producing much poorer malt. We do require nitrogen in our barley. One of the features of nitrogen is that the more nitrogen we have, the less space there is for starch or carbohydrate, so the extract yield of a higher nitrogen barley is lower. Similarly, we need some nitrogen for enzymes. The natural malt enzymes are made from nitrogen, so we need nitrogen available for there. We need nitrogen for foam positive proteins in our beer to give us beer foam. We also need nitrogen for yeast growth as amino nitrogen, and barley will use some of the nitrogen stored in the grain for its own growth during germination. So nitrogen plays an important role. The important thing for us is to control it. If we look at a barley corn, you will need to be able to draw this at some stage during your exams. Quite often this is a question to draw a barley corn, so a labelled grain like this, a longitudinal section of the grain. You can see here we have the chloriza, which is actually going to become the roots. Here we have the acrospire, which ultimately will grow up under the husk, and if left too long will actually break out, and that turns into the leaves of the plant. On the outside we have a husk, and the husk is made up of several layers. The husk here, which is all dead cells, the pericarp and testa, two other also dead cells, but these are semi-permeable membranes preventing sugars and amino acids leaching out of the cell, but also controlling the intake of water and oxygen, because the cell needs to breathe. So very little of it goes through there, most of it goes through the micropyle, and that is basically the outside, what we would call generally the husk, but it's formed of three factions: husk, pericarp and testa. It's in the testa where particularly we find the polyphenols, which are in the cell as antibacteriostatics, but play an important role in haze formation in beer. Under that is this layer of three very important cells called the arylone layer. These cells are where the enzymes are produced, and this is stimulated during growth by the embryo sending out a message of gibberellic acid to activate the arylone layer. And at this stage, these are living cells, and they produce enzymes. Under that, the whole of this area is called the endosperm, and in the endosperm we have the beta-glucan cell walls surrounding a protein matrix and the starch. This is basically the larder, the energy store for the growth of the plant. So we have large and small starch molecules sitting in a protein matrix surrounded by beta-glucan cell walls. And this is an electron micrograph to show the same thing. So here's the embryo. You can see the little rootlets starting to come out, and the acrospire has grown quite well, so it's already started germinating quite a lot. Here's the great bulk of endosperm where the starch molecules are, and the pink is showing you the beta-glucan cell walls surrounding each one of those cells, and that is what we have to disaggregate or break down during the malting process. Underneath this slightly lighter pink layer is the arylone layer, which is going to produce the enzymes which are going to work through the grain, breaking down the cell structure. And on the outside here we have the husk, which is kind of been destroyed in preparing the sample. If we look at the grain, we can see we have a lamella, the upper side, the pellia, the lower side, the husk and pericarp, which we talked about 10% of the grain, and the husk is made up mainly of cellulose along with other hemicellulose, so it's a fairly hard protective layer. It's full of dead cells, there's nothing there, and it has lignin, which is a sort of cellulose type product, phenols, which we talked about, hemicellulases, which are beta-glucans and pentosans, amino acids. The grain is very important both in malting and brewing. In malting, it protects the cereal from being damaged during the malting process, and for brewers, we use the husk of the grain to allow us to have a filter bed through which we can filter the wort. Inside the husk, there's the pericarp, which is the remains of the ovary tightly bound to the tester. The pericarp is dead cells and has no starch associated with it in the mature grain. The tester is a thin double layer covering the whole grain, and both of these are barriers to oxygen and water. The grain does have a micropyle, which we'll see later on, which is a little hole near the embryo, and this is where water and CO2 and oxygen is generally taken up. And this is the main way water is taken into the cell. If we look at the embryo, we can see, as we saw before, the rootlet areas here and here, and the leaf area, or the acrospire here. And these are the areas where the grain is going to grow. So during germination, this grain grows, and initially it grows using a tiny food reserves available within the grain itself. But very quickly those are exhausted, so the grain produces a hormone called gibberellic acid, which passes through this area here called the scutellum and onto the arylone layers along the back here, where they actually activate them and produce enzymes, which then start breaking down the cell walls here and releasing nutrients, which can then be brought back through the scutellum and into the grain to feed the grain itself. Underneath, as we talked about, this triploid layer of cells called the arylone, up to 10% of the grain weight. They sit above the starchy endosperm, and they're living cells which actually have synthetic powers, i.e. they're able to produce and to release enzymes in the barley grain, which are necessary for both the breakdown of the barley grain structure during malting, but also these enzymes persist into the finished malt and are used by us in the mash conversion process. If we look at the arylone layer again, you can see this is sitting under the husk, between the husk and the endosperm, these three levels of cell which are living and actively produce hormones and enzymes. The embryo is where the grain is going to grow, and the scutellum is the barrier between the enzyme and between the embryo and the endosperm, where all the starch is, and so everything has to pass through the scutellum. The last bit that we're going to talk about is the nutrient reserve in the grain, the starchy endosperm. This makes up about 75% of the grain weight, and it is mainly composed of starch. It has cell walls of carbohydrate, mainly beta-glucan, and about 9% protein. These are all dead cells, and basically this is where the starch is contained. If we look at the individual cell, it probably shows us more clearly what's going on. You see this beta-glucan cell wall here surrounding the starchy endosperm here, the starch cells sitting here, large and small starch cells, all sitting on a base of protein. And this is where most of the nutrients in the cell come from. If you were asked to do a question on draw a barley corn and state the function of the individual structures within the grain, this would be a very good way of answering the second half in a table. So here you have husk, protects the embryo during handling and forms a bed during mash separation. The pericarp tester helps the water move through the grain, so a lot of the water actually is transmitted around the top and the bottom of the grain and is slightly slower going through the middle of the grain. And the tester is a semi-permeable membrane, preventing the loss of sugars and amino acids and the attack by microorganisms. The micropyle, which we'll see again later, is an area which allows water to come into the grain, so this is much more permeable, and through which the rootlets ultimately grow. When the germ grows, that the rootlets burst out of the grain and they come out through the micropyle. The embryo, the living structure, which is basically the living seed, which takes up water and nutrient and then will grow into a plant if we give it enough time, or at least will control the breakdown of the endosperm for us. The scutellum, that bit that separates the embryo from the endosperm, we've called it the placenta, which allows water and the hormones to pass from one end to the other, and the nutrients to come back from the endosperm. Areolone, which is this three-layer living cells stimulated by gibberellic acid to produce the enzymes or release the enzymes necessary to break down the endosperm structure. And the endosperm structure is basically the energy store full of the starch and surrounded by beta-glucan cell walls, and it's the enzymes released by the areolone layer which is actually going to work and break down the endosperm structure. So why is barley used for brewing? Well, traditionally barley is not actually eaten by many people, so it has limited food value and therefore was not affecting human nutrition when food was short. It produces beer. We've got used to drinking beer made from barley. Another feature of it has a very large husk, and that husk has been very successful in forming a filter bed for mash separation, and also, since a lot of the malting process the grain is wet, it helps to protect the grain against damage. It's grown very widely, certainly in Europe. One could say it's grown from basically Finland right through to the edge of North Africa, such as Morocco and Ethiopia. It has good enzyme potential and therefore helps us not only to break down all the starch in the malt, but allows us to use adjuncts to break down additional material. In the field, the barley would just grow, take over 2 to 4 weeks, slowly develop, and that will be the natural way. In malting, of course, we want to do it a lot faster, and we don't actually want to grow a seed. So the malting process takes the first stage of germination to break down the structure within the grain, but to leave the starch intact. As we buy our malt, we get two things. We get accessible starch, and we also get the enzymes that we're going to need in our mash conversion vessel to actually break that starch down into sugars. If we look at a longitudinal grain section again, we can see the embryo here, the endosperm, and the 3 levels of husk, the husk, the pericarp, and the tester. And here is the micropyle, and the water comes in through the micropyle. As the grain hydrates, then slowly it depletes, it becomes active at about above 32% moisture, and it starts to grow, initially using a very limited amount of food available within the grain itself, the embryo itself, and then requiring more nutrients. So it sends out a hormone, gibberellic acid, to activate these areolone layer cells, and then the areolone layer produces enzymes which slowly disaggregate, go through the grain, breaking down the cell wall structure, some of the protein, and taking away a small amount of small starch granules, leaving most of the large starch granules intact. And so we can see slowly the modification will work through. The important step at controlling the modification is in fact the water uptake, and you can see the bulk of the water goes in through the micropyle, hydrates the embryo first, and then slowly works through the grain, particularly on the dorsal side, more water passes through the dorsal side than the ventral side, and the central side is where the water is probably slowest passing through. The ease with which that water will pass through will depend on the endosperm structure, and hence mealy grains will hydrate and modify much more easily than steely grains. If we go through the system, then water enters the grain through the micropyle to hydrate the embryo. Water is distributed through the grain by capillary action, and we're basically raising the water content from 12%, which is the typical moisture at storage of the grain, going up to 45% by the end of steeping. Once the embryo reaches round about 32%, then it starts to produce these gibberellins, which pass through the scutellum and activate the arylone layer. And the arylone layer then either releases enzymes or produces new enzymes from de novo synthesis, and starts this process of breaking down the whole of the grain structure in a process called modification. And then the embryo accepts the released sugars and amino acids that have been liberated through the release of nutrients within the endosperm by the enzymes synthesized by the arylone layer. Because that's a process that has to flow through the grain, you can see that the gibberellic acid activates the arylone layer, and that modification occurs in a pattern. So the first bit is nearest the scutellum and the embryo, and the last bit to be modified is the furthest away bit. In well-modified malt, we require the whole process, the whole of the grain, all the beta-glucan cell walls, to be reduced. When we look at the enzymes, we see that there are already enzymes present in the barley. These are beta-amylase, carboxypeptidase, and beta-glucan solubilase. And so as the grain is hydrated and there's a small amount of activity from the hormone and the enzymes, then these first enzymes are released in the first day or so of germination. And these enzymes are particularly important. Carboxypeptidase will break down some protein to amino acid, and beta-amylase is particularly effective at breaking down small starch granules. And so we're getting sugars and proteins being liberated and taken back to the embryo. The next group of enzymes that come through are the gums. These are the ones that attack the beta-glucan cell walls, which are gummy products. So we've got beta-glucanases, pentasinases, which are also in the cell wall, phytase, and beta-glucan solubilase. So all this is going to help to disaggregate the structure of the cell. Alpha amylase appears on the 2nd to 4th day, limit dextrinase on the 4th day. They actually don't play such a big role in germination, in liberating nutrients to germination. Endopeptidase will have a slightly bigger role in helping to release more amino acids. So if we look what we're trying to do in malting, we want to have a limited germination of the grain to produce as a uniform product as possible, the breakdown all the way through of these cell wall structures, the beta-glucan cell walls within the endosperm surrounding the starch granules. And this gives a friable or easily crushable material. When we take the beta-glucan cell walls out from inside the grain, it becomes much more crunchy and soft. And in fact, we use that, as we'll see in the third lecture, for actually measuring the modification, the degree to which the whole cell structure has been broken down. We also need to make sure we get the right balance of enzymes, and these are not only produced but preserved for use by us in the mash conversion. If we over-modify, if we let the process go on too long, effectively we're breaking down sugars that we don't, starches to sugars, which we don't want to do, and we get too much respiration and rootlet formation and lose some of the valuable extract that we require from the malt. If we look at the barley growth here, you can see the barley starts to grow, the little rootlets come out, the acrospire starts to grow, and this will have modified. So the cell structure here would have been broken down. This bit, the bulk of it, would remain exactly the same as barley. After maybe 2 to 3 days, the rootlets have grown stronger, the acrospire has grown up, and now we've got about half to three-quarters of the grain modified. And then maybe just before the kilning, we've actually modified the whole grain right up to the end. The rootlets are at their longest, and the acrospire is three-quarters to completely to the end of the grain, and that's all well modified. And if we look at that in terms of the side of the grain, we can see the first bit, it starts to modify just next to the scutellum, then it starts to come in again, emphasizing that it's modifying nearer the acrospire, nearer the bottom of the grain. The center bit is slowest to convert. Then it ends up here. This is partially modified malt, less well modified malt. It's typically what would be used by some lager brewers, particularly in Germany. So we've still got some unmodified grain here, and the bulk of the grain is modified. It also modifies more on the dorsal than the ventral side. So here we are, we can see the modification occurring here on the dorsal side, and ultimately the bit that's unmodified is that bit in the center. Looking at electron micrographs, we can see that here's unmodified grain, lots of large starch granules, lots of small starch granules, and here's where the beta-glucan cell walls surround the grain. So that would be a typical endosperm cell with large and small starch granules sitting in a matrix of protein surrounded by beta-glucan cell walls and lots of small starch granules. When it comes to modified malt, well-modified malt, you can see here all the beta-glucan cell walls have gone away. Most of the small starch granules have been broken down and utilized as an energy source for the growing grain, leaving us with the large starch granules, which is what we want for mashing. So that's ideal. If we leave it to cook for too long, if we over-modify it, then here we have the breakdown of some of the larger starch granules and effectively loss of extract. So the extract yield in over-modified grain is lower. Factors which affect the modification of the temperature: the warmer the temperature, the faster it will occur, but also that at higher temperature you tend to get a less even modification, and this can cause a problem for brewing later on. And the other thing is, the first stage of the malting process involves steeping, or raising the moisture content from 12 to 45%. And it's been found that instead of keeping it underwater the whole time, a process called an interrupted steep is used, where the grain has a period of wet underwater, and then the water's drained away, and we have then a period of dry to allow it to reabsorb oxygen and continue the active process of taking up water through the micropyle. Under these circumstances, we get modification a lot faster. And ultimately, the other factor that affects it is the ease through which the water can pass through the grain, and that means that steely malt is very tightly packed and will produce very, very poor malting barley compared to mealy, which is open and easy to process. The difference between steely and mealy can be a function of the climate, the sort of growing season. In dry years we're tending to get more steely grain than mealy grain, but it's also very much a genetic process that different varieties will tend to be more mealy or steely, hence the selection of only certain varieties for malting. And lastly, nitrogen. High nitrogen barleys tend to be much more steely than mealy. If we look at that here, we see that if we've got poor modification, that steely grain, they'll have slow water uptake and slow modification. If we have mealy grain with low nitrogen, then we get faster water uptake, faster modification. Another feature which will also affect the modification is the presence of beta-glucan and the enzyme and the amount of enzymes beta-glucanase, which will break down the beta-glucan. And this is again generally varietal. So barley with high beta-glucan, thick cell walls, and low enzyme activity will not modify very well, whereas a grain with low beta-glucan, thin beta-glucan cell walls, and plenty of beta-glucanase activity will modify easily. Just to recap, to look at the mealy grain, this open structure which would allow water to pour through easily and break down these beta-glucan cell walls, and the steely, the high nitrogen, very tightly packed grain which will modify very, very poorly. So in steeping, when it comes to steeping, we look at temperature of the steep water, which is important, the grain size, the evenness of grain size, the protein content, and that's a specification on our barley. We will specify that we want a protein between 1.5 and 1.8 or something like that to avoid having steely grains. The viability, in order for the grain to actually germinate, it has to be alive, so it's very important that we have only living grains there. Possibly seasonal effects of crop year. We can get split grains, which we'll see later on. We can get high nitrogen barleys if it's low rainfall. The variety is very important. Things like this affecting the beta-glucan cell walls and the ability to produce beta-glucanase enzymes, and the steeping regime, which is altered throughout the season to compensate for dormancy, because as we'll see later, grains don't immediately germinate as soon as they're harvested, but have an inbuilt dormancy. If we look at grain size, for example, the effect of temperature, we can see that obviously at colder temperature the water uptake is much slower, so warm water will be taken up more quickly, and typically steeping is between 15 and 20 degrees Celsius. It will be more precise, but it depends on how difficult the barley is to malt. The harder it is to absorb water, the harder it is to malt, the earlier in the season, the higher is likely to be the steeping temperature. The other issue we talked about was the grain size, and we'll see here in the next slide. It seems to have frozen for a second. The effect of grain size, and small grains take up water a great deal faster than big grains. So here the small grains taking up water very fast, the big grains, once they get above 2.2, it's much flatter. And so we would have a specification saying that we want 90% of our grains greater than 2.2, or 95% of our grains greater than 2.2. So we would specify the grain size to ensure that we had flat process on the graph, and therefore we take up water evenly throughout the hydration during steeping, because if we have uneven water uptake, we'll get uneven germination and hence uneven barley. In some countries where there are a lot of smaller grains, these are often screened out, and the two samples are actually malted separately. So you have here the bigger grains malted as premium malt to produce premium malt, and the smaller grains malted to produce second standard malt. These may even be mixed together at delivery, but generally in most countries the barley corn size is round about 2.5 and is fairly consistent. Before, when we have the barley delivered off the field from the farm coming into the maltings, it's absolutely important to take a sample so that we can then check all these parameters. And because it's a bulk solid, it's difficult to get a representative sample, so we actually have sampling sites here where we take a probe down and sample in a pattern across the grain to get a representative sample. When the barley comes into the maltings, we now have to evaluate it to make sure that it is suitable for malting. The first thing is to look at it by appearance. So we look at the sample we've got, We look at the whole grain. We don't want broken grains because broken grains can lead to microbial infection. We like the grain varieties we prefer are wrinkled grains, grains with good wrinkle husks, because that actually helps water distribution and improves the degree of hydration of the grain during steeping. The grain should be sort of light straw coloured. Discoloration indicates fungus, particularly pink grains, which are often associated with a fungus called fusarium. And we may cut the grain open to look at it to ensure that it's mealy and it's got a nice open structure, flowery looking. If we cut it open and it's steely, it looks like steel, shiny, and we know that it's tightly packed and will produce poor quality malt. Here we look at size, and for example, we want 98% greater than between or 2.25 and 2.5 for steeping and milling. If we have different size grains when it comes to milling, it's very difficult to set the mill roller gap to crush all the grains equally. We would smell it. This is a good way of working out if there's any mold on it, and we would be looking at it to make sure that there are no insects crawling around, because insects will soon grow. We want the right variety. We actually find that we malt barley by variety. You can't malt a mixed variety of barleys because they will all behave differently during steeping and germination. And we don't want other seeds present. The most important, or one of the most important things we look for is the absence of fungus, because fungus can leave behind, although it won't survive the malting process, it can leave behind mycotoxins, which are poisonous to humans at fairly low levels. And also, if there's a fungus there, it may also lead to gushing, which is the spontaneous expulsion of beer in the finished package. And so this is a very important HACCP control point, HACCP control point, where the grains are individually inspected to ensure there's no fungus present in the grain. And lastly, we check for moisture. We want to store our grain. We have to store our grain for a year, around about 12% moisture, so we're going to have to dry it down from field moisture. But if we're looking at 15.5% max off the field. If it's above that, then we actually charge a premium to the farmer and take some money off to compensate for the higher water level. So again, here's the hand evaluation: appearance, as we said, smell, moisture and dampness, shape and size of grain that can be used to help identify that it is the variety we're expecting, husk properties, thickness, wrinkling of the grain helps us again with varietal identification, uniformity for blending, making sure it's free from grain, stones, straws, insects, dust, damage, that the grain isn't damaged, it isn't broken, and there's no pests or fungi or holes where things have already eaten the grain. In looking at samples here, you can see this is one with a split grain. This wouldn't be any use because it's lost its viability, it won't grow. You can see already being prone to fungal attack here. Here's another sample where we have different grains, so they're different varieties here, the purpley one and the yellowy one, and so we would have to separate them out because we couldn't malt them separately, and therefore we need to send that load away. Here are grains that have started to germinate, pre-germination. You can see that it's actually broken out here. You've got the rootlets here and the stems starting to grow, and this is an example again showing you how the acrospire is growing up, and these grains, certainly at the bottom here, are dead, will not survive the malting process, whereas these grains here will germinate. The stain, we'll see in a minute, called tetrazolium chloride, is used as a rapid method for looking at viability of the grain. So it's ideal for grain viability to make sure that what we're going to steep is going to be alive and well. And here we look at what the buyer wants. This is what we'd ideally want to be delivered to our maltings to make good quality malt. Nice even grains, nice wrinkly husk, all whole, no broken grains, no broken damage to the grain, and that would represent a good sample of barley if it's the right variety for malting. We need to watch out for our pests, these type of insects, grain weevil, red grain beetle, sawtooth beetle. You don't need to know them. You need to know about pests. You don't need to know anything about the individual insects involved. Then we start doing some analytical work. The first thing is we can grade the grains to make sure that the sizes are correct. So here's a 2.5 screen, 2.25, 2, and 2.2 and 2. And we put the grains in here, there's riddles them, shakes them up, and then looks at the amount that is retained on each one of these screens. Another way of doing it is by weight, such as 1,000 corn weight or a hectolitre weight, but typically screening is used to ensure that most of our grains, 95%, are greater than 2.5. The other thing we may do is cut the grain open and possibly look at it to see if it is either steely or mealy. So there we are, assessing the grains, looking at the number that were passed through the screen. The sizes will depend on years and depend on the type of and variety of barley purchase. So here we're talking about 90% of the corns greater than 2.5 and less than 5% smaller than 2.2. So again, we want even germination from even sized corns. If we have lots of small corns, we'll get uneven germination. If we have lots of small corns, we have to discard them. That leads to higher malting and potentially brewing losses. And again, we need to be able to set our mill gaps around the corn size. We've mentioned protein or nitrogen. Nitrogen and protein are related. 1% nitrogen is equivalent to 6.25% protein. So in IOB methods, we quote our malt in nitrogen 1.6 to 1.8, whereas in EBC is quoted as percent protein, which is where we've multiplied it by 6.25, and it'll be somewhere around about 10.2 to 12 or something of that sort. We pay a premium for barley with low nitrogen, and we'll see that obviously the higher the nitrogen, the less extract we get, because it's actually competing with the carbohydrate, the starch, so we get less starch and more nitrogen, so we get lower extract yields. But of course, in some years where there's low rainfall, quite often we have to deal with barleys that have higher nitrogen because they grow less carbohydrate. We can see it varies for different varieties. This variety is showing you the fall off of extract. This is extract in litre degrees per kilogram, or could be 75, 80 Plato, going down to say 70 Plato, so it's falling down in strength as the nitrogen levels increase. But that is a good variety, a good malting variety. This variety here, the fall off is much faster, and it's quite probable that this is starting to become a little bit more steely, and hence is going to modify much less well. We measure nitrogen and moisture on the same instrument nowadays, near infrared, at 2 different wavelengths, and these are all calibrated online, so there's a network where all the instruments are calibrated weekly online to ensure that everything is working correctly, because it's quite a fragile measurement. It's nice and easy to use and very quick in results. The other thing we need to do when the barley comes in is look at its germinative capacity. So is it alive or is it dead? And we test that. 2 other tests that are done on barley that are relevant are germinative energy and water sensitivity. Germinative energy and water sensitivity are only done when the barley is going to be steeped. So barley will come in and we may keep it for up to 6 to 9 months or even up to 14 months before we need to steep it in order to produce malt for our customers. So again, we don't need to have the germinative energy or water sensitivity until we steep it. And germinative energy is a measure of dormancy. We'll discuss that slightly later on. So germinative capacity is the percentage of corns which will germinate. The traditional way is to take 200 corns and steep them in hydrogen peroxide. Hydrogen peroxide releases oxygen and therefore overcomes dormancy, so it compensates for dormancy. And here we can see it, here the grain steeped in hydrogen peroxide, and then after 3 days we take it out and we see how many have grown, how many little chits have been produced, and that gives us a measure of viability. And we would want to buy barley that has got either greater than 96 or 98% viable, because obviously barley that's not alive will not germinate and therefore will stay barley throughout the malting process. On the other hand, this is a slow test, so there is the rapid test that we saw earlier, and that is staining the grain with this salt, tetrazolium chloride, and it stains pink, as we'll see, and only the living grains take up the pink colour. The dead grains will stay white. So it's a rapid way of measuring it. Here's the living grain here, so those are viable, pink, and these are not viable. The problem comes these intermediate grains here, and it will be an assessment by the maltster whether that which has started to germinate, the pre-germination, whether these would be considered living or dead. Generally that's not found most years. This is a chart put up for maltsters. So in the nature, the barley has dormancy, so basically the barley is harvested in July and August, but we actually don't start to try and steep it to make malt out of it until end of October, early November at the earliest. So it has an inbuilt dormancy. We often want to be able to break that dormancy, and the way we test for that dormancy is germinative capacity. So there we are, the grain is dormant, but we can actually measure its viability with the peroxide test. But if we actually tried to steep it in water, we'd find that very few of the grains would germinate. Similarly, if we put it very deep in water, it will be water sensitive. So in other words, it can't get enough oxygen to grow, and so it will not grow. Its performance, its germination level will be even less. So how do we overcome dormancy? So if we want to break dormancy, we can actually heat it up. Warm storage will help to break dormancy, and also things like adding additional hormone, this gibberellic acid, will also help to break dormancy. So if we look at what typically happens, barley off the field will not germinate, hardly germinates at all. So this is a germination test. We do a test at 4 mils and a test at 8 mils to see the percentage that will germinate before we steep it. So this barley would not germinate, would not produce malt. In this case, it's actually broken dormancy in this partially matured, but it's very, very sensitive to water. So we'd have to develop a very special steeping regime where we allow plenty of oxygen in, and that's how we do it in these interrupted steeps with periods of submersion underwater followed by air rests. And so this would be a difficult barley to steep. On the other hand, this barley, which is mature, has now overcome both the dormancy and the water sensitivity, and this barley would be ready for steeping, and we could find a much longer wet period, but we would still have an interrupted steep to maximize the water uptake. So the germinative energy, how we work that out is to take 100 grains and place them on top of a filter in a Petri dish, and actually measure the number of chits actually made. And water sensitivity, a similar test, but this time we use 8 mils of water, and this is enough to cover the grain, so it's underwater. So if it isn't able to get enough oxygen, it will restrict germination. And so we look at both tests. To see this most easily, here is a typical test being done in the laboratory. This is the 4 mil test where the grain sits there and is only partially covered by water. We see how many chits are produced. And this is the 8 mil test where the grains are completely covered by water, and we see how much it is affected by water. And based on those two tests, we can work out, first of all, if the grain has broken dormancy and can actually be malted, and the second, on the 8 mil test, will help us decide what type of steeping regime we would use in order to get the optimum amount of malt out of it. We can see that temperature helps us recover from dormancy, so if we kept our barley cool, it would stay dormant for much, much longer. If we actually let it warm up, then dormancy breaks much more quickly. And if we actually dry it, which is what we have to do to get rid of the moisture, if we dry it, here we can actually break dormancy. So barley drying not only is used to take away the moisture, but is also important in actually reducing the amount of dormancy in grain. Before we malt the barley, we do a micro malting trial, because we will check that the conditions that we've set up are suitable, or if we've got new varieties coming through or other things, we have this micro maltings trial where instead of malting, say, big 500-ton batches, we can malt single 1 kilogram batches and take them through the steeping, germination and kilning regimes and simulate those in the laboratory to check that the conditions we've decided on are suitable. We've also got to look at contaminants. This is on barley intake. So we've talked about the fungi, fusarium, and other fungi like penicillium that produce mycotoxins, and we'll see this at the end, that these are critical from a hazard point of view from health, and the maltster's got to ensure that all the barley taken into the malting are free from these organisms. Pesticide residue is also a concern, and the barley actually has a passport when it's delivered, and that passport is made by the farmer, who states what pesticide he's sprayed on and when, and there are obviously permitted times and pesticides which can be used. Heavy metals, generally not a problem, but it could be depending on the pesticide residues and also the soil. And the other one is genetically modified material. Again, very little barley is genetically modified, and we're going to talk about that in a minute. But some companies specifically don't want to use genetically modified material in their products, and so it is prescribed by a number of major brewing companies. When we come to evaluate the barley for malting, we make sure that it can take up the water adequately. This is partly the structure and also the way we set up our periods of wet and dry, our interrupted steep. We make sure the barley is a mealy grain, so it's the right variety, and in the season it's low nitrogen, so that it will actually be able to hydrate evenly and efficiently. We want to make sure that it's alive, germinative capacity, that's the tetrazolium chloride or the hydrogen peroxide test. And then when we come to steep it, that it's broken dormancy, the 4 mil test, and we take account of its water sensitivity, how likely it is to drown, and that is the 8 mil test, and we just check the grain to make sure there's been no pre-germination during storage. Here's a typical barley plant, so one corn will produce 2 or 3 tillers, and in each ear there will be 15 to 35 grains. So that's what we're going to harvest, both for seed and also for malting. Over time, we've been doing a lot of barley breeding to produce better barleys, Specifically, the aims in this sort of thing are yields to the farmer to get more higher yield per acre. Disease resistant, this will reduce the cost of using pesticides and fungicides, and similarly reduce the amount used, so it's a win-win situation for both of us. Agronomic characteristics, barleys that perhaps can withstand drought, for global warming, or higher temperatures, that can withstand wind tolerance, i.e. short stalks, which means it doesn't blow over so easily. And last but certainly not least, barleys that make very good quality malt. So over time, the yield to the farmer's gone up about 1% in the last 50 years. We get better disease resistance, and there is a new emphasis on grain quality. So the quality of barleys provided today as a result of breeding programs are far superior than those that were available previously. The way we can do it is selection by selecting the best looking grain, or the grain that is most suitable, directly, by hybridization, by breeding grains to achieve the correct character, or by genetic manipulation. We can either cause or accelerate the mutation, which is a change in the genetic code within the grain, or most specifically in genetic engineering, introduce a gene from another organism which confers a particular advantage to the grain. In terms of crossbreeding, here we would look at hybridization, where we would actually, the barley is mainly self-fertilized, so the anther, the pollen from the same plant will stimulate the anthers from the other plant, and therefore this will actually self-fertilize. To avoid that, we have to take off the male parts, the pollen parts, and let it remove all the anthers, so let it self-fertilize. And here are examples where they're covered up to prevent them being fertilized from a neighboring plant, so that we can only put up exactly what we want and crossbreed it with the pollen from the plant that we actually want to breed. And typically, first of all, in selection, we might go through a series of barleys and look at them and say, yeah, that would be the best one to use if we were just selecting a barley. If we wanted a breeding program, we would look at these two and say, yeah, there's an advantage in using breeding that barley with that barley, because we'll get fatter corns and longer stalks, or disease resistance. So we'll crossbreed, and that's where hybridization occurs. Here's a barley there with the anthers where the pollen is, and the stigmas which are fertilized and go down to the ovules. So in a breeding program, we'd remove the anthers and we'd actually add the pollen that we require onto the stigma here, so that we actually produce the right seeds, the right crossbreed that we require. And it's a very specialist process. It's also a very time-consuming process. It takes about 12 years for a trial from the start of the breeding program. Here you are, F0, the parents, up to the final product. And of course it goes through a variety of steps. The first step, the F1 generation, are all similar, and then we start seeing huge variations, and we have to select those that give the characteristics that we look at. So as we breed it forward, we suddenly start to see the variations start appearing. In the UK trial system, we start off having to get approval. We start the breeding program, and when we have enough seed, we go to the national agricultural body, which then puts it onto a recommended list after 3 years. And once they're there, we can then start growing those, and as we grow those up, we then take them along to the maltsters and the brewers through the Institute of Brewing Malt and Barley Committee, and check that the quality of that grain is adequate. Here we have the different plants, individual plants here, 2 million plants, and then slowly we whittle them down to fewer and fewer. seeds as we select only those that are suitable. So you can see again this 12-year process, and it takes a long time before we can actually come up with seeds that are recommended. And here's a typical patch, sort of test strips, where the different seed varieties are being checked and grown on, so we can get sufficient samples for testing and identify those that are healthy and vigorous and those that do not meet the requirements. In genetic engineering, what we're doing is basically interfering with the genetic code, the DNA, within the cereal. And there are two principal ways we can do it. One is by mutation, by using X-rays or gamma rays, or chemicals that will come along and actually change the base sequences. Now this is much more structured, and we have special targeted ways of changing particular bases, and now that we have the genome of most organisms, we can work out which bases we want to change to affect an improvement. And varieties that have come up with from this route include low anthocyanidin malt, and also a new malt that's called a null lox, a malt that doesn't produce the enzyme lipoxygenase, and we'll be discussing that in the second section. So that's one way of doing it, and in some countries this is not viewed as genetic engineering. In other countries, again, genetic engineering is basically discouraged, and some brewery companies will not use materials that have been genetically engineered. And genetic engineering generally is where we've taken a gene or a series of bases from one organism, and we're going to implant it in another organism. One of the ways was to select the gene directly, put it onto little gold particles, and fire it into the plant tissue. So that's one of the ways of doing it. Another way is where we can transfer it through a virus, a phage, and basically we get the gene into the phage, and then when it goes across to the new barley corn or the barley nucleus, the new DNA is incorporated in, and the new plant acquires the attributes. Things that we're wanting is yield. We want higher extract yield. We want standing power or short straws to prevent it lodging or falling over. Early ripening, so rapid growing of the grain, is preferred. Winter hardiness, being able to overcome colder weather in the winter, and the latest safe sowing date, again that's coming back to early ripening, and obviously also a number of malting characteristics which we'll deal with in a minute. For any variety to go forward, it has to meet the DUS test. It's got to be distinctive, different from any other variety, so it has to have certain differences. It's got to be uniform. The variety has got to be pure, and therefore all the grains have got to be similar. And the last thing, it must be genetically stable. So if we introduce a gene or do a breeding, we don't want it to backbreed. It's got to stay the same variety over generations. Because we expect lower nitrogen and lower yield in the field, so the farmer gets less yield from growing malting barley than he does from growing feed barley, we have to pay a slightly higher premium for malting barley. Variety is also important. So malting, we must have the right varieties. We want to malt them all together, and certain varieties are good for malting, other varieties are bad. This is varieties that are very steely or have high beta-glucan cell walls or low beta-glucanase enzymes, those type of things. To be able to identify a barley variety is quite difficult. So we actually can physically look at the grain, look at certain characteristics, the gape, the outside, here you are, the pellier, the size of the pellier. One of the easiest ways to describe it, if you have a question on it, is look at rachilla. This is this little stem part here, and you look at the hairs and the length of the rachilla. We can also use the back of the grain, the dorsal area of the grain, to look at the wrinkling and the hairs here, and the base, the lodule here, and the colour, and various things like that, which help us to decide which variety it is. A simple way here is a rachilla, and this one is one variety. It's got a short rachilla, very short rachilla, but they have long rachilla hairs, and another barley variety here has a long rachilla with short hairs. So you could easily, with a hand lens, tell the difference between the two barley varieties, and there are tables which show you the particular characteristics of each barley variety. That was the old-fashioned way of doing it. Few people use that now, and most people now use gel electrophoresis, where we actually use chromatography to separate out the protein fractions, and each barley will have different protein fractions. Not only can we tell whether they're different varieties, but we can also see quite easily whether they're related or not by the amount of similarity in the barley and the protein fractions. So this is a rapid and effective way of looking at barley varieties. Lastly, what have we achieved? Well, from the farmer's point of view in breeding, he wants a higher yield, more tons per acre, good straw so it won't lodge, good short straws. He wants the barley to all become mature and rapid maturation. He wants it all to germinate when he comes to plant it. He wants it all even so it gets top grading, and he needs to meet our protein content, so he needs to be between 1.4 and 1.8 in order to sell the barley. He obviously wants disease resistance. This is no longer part of the syllabus, but to know that we want certain disease resistance is important. But when it comes to us as brewers, what do we want? We want fast modification, good mealy grain to allow easy transfer of water and enzymes, so it all modifies. We get well modified malt. We want high extract yields, plenty of starch. We want low viscosity, low gums, so we want to be able to break down the beta-glucans here as effectively as possible. So we want barleys with relatively low beta-glucan to start off with, and plenty of beta-glucanase, and that also affects filtration. Viscosity and filtration are closely linked, and high viscous beers or worts are difficult to filter. We want plenty of enzyme power. That's largely controlled by the maltster. Barleys will produce the right enzyme power in general. We want to be able to break down the protein easily, so the protein's got to be of the right form, and that's important in making beer malt colours and the beer foam, and we want it to be fermentable. So the outcome of that is fermentable, so that's low levels of small starch granules, high levels of large starch granules. We've also got to check on these moulds, and we can see that during storage, in some instances, the moulds will increase, or the mycotoxins produced by the moulds will increase. So in this case, the mould is increasing after storage and increases slightly in the malt, but that's probably due to drying. So this has gone from 12% to 5%, so the increase could be as a percentage of the drying, whereas in some toxins, for example, they fall, but again, there's this difference between the barley and the malt due to drying. So again, we have to make sure that we don't have too many moulds or any moulds on our barley because of the health hazard, the HACCP point. And that basically concludes the lecture on barley.